Good afternoon. This is Dr. Famuyiwa, and today I'm going to be talking about some genetic testing, PGT-M. I hope you find it useful. The impetus for doing this talk came from my patients who've always asked me, what is genetic testing? What is PGT? Why should we do it? What will it do for us? Well, PGT is pre-implantation genetic testing. Why would you do it? Because in today's world, no one should be blindsided and have an affected embryo that's not viable without knowing about it. So basically, we are going to test the embryos before they are transferred into a uterus. In order to do this, let's talk about how do you even form an embryo? How do you form a zygote? How do you fertilize an egg? What happens is the sperm typically travels up through the uterus, up into the fallopian tube, almost like a heat-seeking missile looking for the egg. It's kind of tunneling right to. When that sperm meets the egg, right, typically happens in the fallopian tube, the sperm releases a chemical called the acrosome that tunnels a hole through the top layer of the egg, okay? So the sperm has this translucent cap on here called the acrosome that contains ingredients, chemicals, that can drill a hole through the surface layer of the egg. Now, what happens is once one egg penetrates this surface layer, there is a reaction by the egg called the cortical reaction that hardens the surface of the egg and prevents other sperm from getting in. Now, once we have the egg and the sperm, we're going to talk about fertilizing them, mixing the DNA up. Now, understand the sperm and the egg are composed of half the genetic material of a full-grown individual. It's when the egg and the sperm meet together that you now form a fully formed individual that has the full complement of DNA, 46 chromosomes. So you have the initial zygote being formed. Now what happens is, let's say you have the egg here. Once the sperm penetrates, the DNA of the egg and the sperm first line up together to exchange genetic material. You form a zygote. Immediately, this zygote starts to divide. So you have first 2 cells, and then a cell, and then you have 8 cells. That's the zygote. I just showed you what happens with the egg being fertilized and forming the embryo, which is initially called a blast. What I'm going to show you now is, where does this happen? Well, it actually happens in the fallopian tube. So the egg releases the ovary, ovulated egg. The egg is picked up by the fallopian tube. It meets the sperm inside the fallopian tube. So sperm fertilizes the egg in the fallopian tube. Right away, you see 2 pronuclei. This is a fertilized egg called a zygote. And it starts to divide. The division I showed you occurs in the fallopian tube. As it's dividing, first into 2 cells, 4 cells, 8 cells, and so on and so forth, the fallopian tube is gradually moving it towards the cavity. By the time it gets to the cavity, it has grown into this structure called the blastocyst. This takes usually about 5 to 7 days. So what happens is, one of the things I want to bring to your attention is, as the blastocyst is formed, it is different from the cells at the 2-cell stage. So how is that? The cells at the 2-cell cells, when they separate, they can form 2 brand new individuals. They are very identical. Unlike the blastocyst cells, the cells here have started to separate to become specialized, if you will. And the part that will become the baby is clustered at the bottom. It's this big structure. The part that will be the afterbirth is on the outside. So this structure is almost like a sphere, right? It looks like a hollow sphere. With the part that will become the baby is clustered on the inside. The part that will become the afterbirth, not contributing to the baby, is going to be on this hollow outside. So now let's see what happened. All right, this is Dr. Famuyiwa. We're continuing our talk on pre-implantation genetic testing, genetic carrier screening, and we left off where we discussed the formation of the blastocyst, the embryo, right? That looks like the structure I'm showing you here. The unique thing about the blastocyst is that the cells, they're committed. The parts that will be the afterbirth is lined up on the outside. You see this yellow group of cells. The whole thing looks like a hollow sphere, and the part that will become the baby is clumped up at the bottom, all right? That's called the inner cell mass. So now we have an embryo that's being created from mommy and dad, right? We know that the DNA to create that embryo that will become baby and little Joey and Jane come half from daddy and half from mommy. But here it lies the crux. Mommy and daddy can also pass on some genetic defects to the babies, right? And the most common one we sometimes see is if they have what is called an autosomal recessive disease, the sickle cell disease, right? So most people from Africa are probably familiar with it because we get tested for hemoglobinopathies and they'll tell you they're AA, AS, or SS, fully meaning full-blown sickle cell. And what happens is mommy has a recessive gene for the sickle cell trait. Daddy has the same recessive gene. When they come together, there's a 25% chance that they will pass on the normal gene, so you have a normal embryo from a normal baby here, or they could both pass on the affected gene where they now have a 25% chance of having an affected child. They also have a 50% chance of having children or babies that are just carriers like themselves. Now, the unique thing is when we're talking about carrier traits, right, on your DNA strand, there is a particular mutation on that strand that's causing the problem, okay? So you look at your DNA helical twist structure, the funny structure you usually see. Maybe there's one tiny little part of it that's not right. That part is sufficient to cause the genetic problem. Now, this is an autosomal recessive pattern, meaning that you need two of those affected genes to cause the disease state. There are other instances where you only need one, and that's called an autosomal dominant disease, such as Huntington's disease. You only need one gene. So if one parent is affected, remember the sperm is reduced or the egg is reduced, one gap, one chromosome they'll form 4 possible combination of gametes. In this case, there's a 50% chance of having an affected child because you only need one copy of the gene. And then there's a 50% case of having an unaffected embryo. Well, how can you find this out? Well, we can do, again, I'm not talking about AS, SS and AA for my, patients who come from Africa. I'm talking about looking for the mutation in the DNA that actually is responsible for this. So you'll get blood sent or you could get a buccal swab where we will send this to a genetics lab and they magnify the DNA looking for the particular gene, the mutation that's causing the disease problem. Now, that's almost like looking for a needle in a haystack. When you find the mutation, so I tell my patients, we got to do your genetic carrier screen first to identify the mutation that's causing the problem. Once we have the mutation, that means we know what it looks like. We can now create a probe to look for that specific mutation. Everybody may have a slightly different mutation. So sometimes the genetics lab may say, well, we're almost there, but we might be able to fine-tune it better if we have additional blood from your siblings or your parents if they're around. So we want to hone it down to see, can we create a mold to look for this particular mutation? That mold is called a probe. It takes 10 to 12 weeks to develop it, especially if the genetics lab has never had to look for that mutation before. Now, if they've created other probes for something similar, they may be able to turn around a lot faster. So please, it takes time to get the probe. Now we have the probe, we have the mold, we know what to look for, we have the right microscope, we know what to see under the microscope. We now need to find out if the embryos created have that needle in the haystack, right? We can't tell by looking at it, but we have this magical probe that will zero in like a heat-seeking missile, looking for that needle somewhere in the DNA of the embryo. So how do we do this? Remember, we have the blastocyst, the part that will be the afterbirth is on the outside, the part that will be the baby is on the inside. So then knowing that, we can take samples of the part that will be the afterbirth. Remember, this embryo is like a sphere. We can take a few cells, just biopsy off a few cells, but leave the part that will be the baby ago. We send these cells off to the lab. They now magnify the DNA of the cells. They gotta magnify to see what they're looking for. Remember, they already created the probe. They now apply their probe to these cells to see if those probes can latch onto something on the cells. In other words, can it find that needle in a haystack? Can it detect the mutation? Once the probe seeks and hits, we now get a report back saying biopsy number 1 from embryo number 1. And by the way, we do have to freeze the embryo while we're waiting for all these test results to come back. Biopsy number 1 has the full-blown disease state. Biopsy number 2 from embryo number 2 is free. Biopsy number 3 from embryo number 3 may be a carrier. Using this information, you are now to go select the embryo that may be unaffected, right? So you can get a healthy baby. So this is Dr. Famuyiwa again. Join us we're going to be opening our own podcast. It will be Fertile Talks. Please be on the lookout wherever you get your podcast from, YouTube, Spotify, and please subscribe, like, and share. And please send us any questions you may have at MontgomeryFertilityCenter.com. We will entertain your questions and create hopefully more enlightening videos for you. Thank you. This is Dr. Famuyiwa.